Do we have signal at this point? Okay, so I'm going to finish up here, and then I'll be right with you. Another exam. It's the summer, so they come around really, really fast. And so that exam will cover everything through Friday. And so what we'll do today is I'd like to get through descending pathways, and then we'll have tomorrow to talk about muscle and interaction between the nervous system and muscle. But where we're going to begin today is I'll show you some slides that we couldn't show yesterday. And then I'm going to remind you about the PCML, also known as the DCML. And we're going to add to that the spinothalamic tract. Now, the DCML and the spinothalamic tract together are considered to be ascending somatosensory pathways. They're ascending because they're headed up the cord, they're headed to the brain. And they're somatosensory, somato meaning body, sensory because of the senses. So this is sensory information coming in from the body. And again, there are these two pathways that we'll talk about, the DCML and then the spinothalamic tract. I remind you from yesterday that the DCML or PCML is carrying information about proprioception and discriminative touch. The next character, which is going to have all the same characteristics that we talked about yesterday, the 3-cell pathway and all of that, is going to be the spinothalamic tract. Again, emphasize this term, spinothalamic. The first syllable, spino, tells you that's where the neuronal cell bodies are, where these neurons are receiving synaptic input. Thalamic tells you where the output regions are. It tells you where these neurons are making synapses. So this will be the spinothalamic tract. And this is nomenclature we'll run into as we go through these systems. Again, it's going to have the same story that we talked about yesterday for the DCML. It's going to have a primary sensory afferent whose cell body is in the dorsal root ganglia. It's going to send processes into the central nervous system. And then it's going to have slightly different story to it. Where yesterday we talked about the primary sensory afferent coming into the spinal cord and ascending the cord on the same side where it entered. What we're going to see here is that the primary sensory afferent comes into the spinal cord, and almost immediately where it comes in to the dorsal horn, it will now make synapses onto second-order neurons. So hold that idea in your head. Yesterday we didn't reach the second-order neuron until we got to the brainstem. We had to send those axons all the way up to the brainstem, to the dorsal column nuclei, where they made the synapse between the primary sensory afferent and second-order neuron. Second-order neuron crossed the midline. That's the rule. Second-order neuron always crosses the midline. So we'll see then another story here where the primary sensory afferent of the spinothalamic tract obeys all those same rules. Never crosses the midline, has the cell body in the dorsal root ganglia, and then sends a process into the central nervous system. Story is the same up to here. But now it changes a little bit. At this point, the second-order neuron synapses onto, at this point, the primary sensory afferent synapses onto the second-order neuron in the dorsal horn of the spinal cord. And then the second-order neuron, remember it always crosses the midline, sends a process that crosses over to the other side of the cord and ascends the cord in something known as the spinothalamic tract. It's a second-order neuron, so it's destined to synapse onto third-order neurons in the thalamus. So the spinothalamic. So it's going to have these second-order neurons are going to synapse onto third-order neurons in the thalamus. Then those third-order neurons are going to send processes to the somatosensory cortex, just as the third-order neurons did yesterday in the DCML. Then I want to spend a little bit of time on Brown-Séquard. Again, as I explained yesterday, if you understand Brown-Séquard, you understand these pathways. And if you can look and explain what happens in Brown-Séquard, you understand what's going on here. So I'll use it as a kind of mechanism to inform you about these two pathways, but more importantly, to show you how severing the cord is going to interrupt some information and not others. Then we're going to start talking about brain structure, because after all, where we're headed with this information is to go all the way up to the cerebral cortex. So we're going to do a very short segment here on brain structure. Now, you can take a neuroanatomy course and you'll spend all semester learning about different pathways and different connections in the brain, different parts of the brain. But we have to get through this fairly quickly. So I'm going to distill it down to the brainstem, where we saw those dorsal column nuclei. The thalamus, which is in fact part of the midbrain, because that's going to be the site where we're going to relay this information from the second order to third order neurons. And then finally to the cerebrum. And again, the cerebrum, we talked about a little bit yesterday, the cerebrum is what you think about largely when you think about the brain. It's only a part of the brain, but we think about it because when we imagine pulling the skull cap off and looking down in, you're looking at the cerebrum. The business end of that structure is, in fact, a very, very thin veneer of about 6 cell body. It's a thin veneer of 6 neuronal cell body layers. It's always the same. We always have 6 layers wherever you look. And that's the cerebral cortex. And that's where all the synaptic integration occurs. That's gray matter. But most of the cerebrum is, in fact, white matter. And the white matter, remember, are the myelinated axons. And those myelinated axons now are sending information to all different parts of the brain. And although we think about the gray matter as being the functional part of the cerebrum because that's where all the synaptic activity is going on, your brain can't work if it doesn't communicate with other parts of the brain. So those white matter connections are absolutely essential for brain function. And we now know that traumatic encephalopathies are often a damage to the white matter, not to the gray matter so much. But if these wires are broken and they can't communicate from one part of the brain to the next, then communication breaks down. So although we'll emphasize the importance of the gray matter and analysis of information in the gray matter, recognize that this is isolated analysis. And the way your brain works is the interconnection of all these analytical points. That then will bring us to the next thing I want to talk about, and that's going to be somatotopy. Somatotopy is a concept that was first discovered in the early 1900s, then rediscovered in humans in the late 1940s. And what it says is that the body plan is mapped out in the organization of the cells in the cortex. So there's going to be a region in the somatosensory cortex that's dedicated to sensory input from the face, another that's dedicated from sensory input from the hand, another from sensory input from the feet. But one of the interesting characteristics of that is that the size of those regions that are mapped out on the cortex is not proportional to their anatomic size, but rather is proportional to the amount of innervation. So we're going to see this very odd-looking, what they call homunculus, that shows how much innervation comes from each part of the body. And we'll talk about that in terms of the somatosensory cortex. Then we'll imagine that we've had all the sensory information come into the somatosensory cortex. And one of the ideas we presented in the very first discussions of the nervous system was that 3-box model: sensory in, integration, motor out. I just want to touch on this a little bit because I don't want to go directly from sensory input to motor out, because in fact there's this important part of the brain called the parietal association cortices. And what they do is they take information in from all sensory modalities. They're taking in information from sound. They're taking information in from sight. They're taking information in from proprioception and somatosensory information. And they're organizing all that information so that you're aware of the world around you. And from there, you make a decision. decide what you're going to do about the organization of the world around you. That information is then sent from the parietal association areas up to the frontal lobes. And in the frontal cortices, there's a whole bunch of integration that goes on, and you make a decision as to what to do. And that decision then is sent largely to the motor cortex, and then that information descends through the motor cortex. Now, if you take another course, we're going to just talk about the motor cortex here, primary motor cortex. If you take another course, you'll see there's a whole bunch of integration among these different premotor areas and the motor areas. And you'll also recognize that there's another different part of the brain called the striatum, which is really, really important in movement. And so, if you take a course in neuroanatomy, you'll stress those points. We don't have time for it here, but I'd like to get through that. And then by the time we get the descending pathways, we're going to talk about the upper motor neurons in the motor cortex, sending processes out into the internal capsule, crossing over the midline and desiccation of the pyramids, and then descending the cord in the lateral corticospinal tract. That's a tract, so it's going to be conductor regions of the axons, or conductor regions of the upper motor neurons, and these conductor regions of the upper motor neurons then will synapse onto lower motor neurons in the ventral horn of the spinal cord. And we'll walk our way through that. But let's take a minute here to pick up some slides that we missed yesterday. So this first image is one that I like, and I wish I could have shown it to you in more detail yesterday, because it gives a better three-dimensional image of the spinal cord than what I could show on the board. And so if you look at this, it's as though I'm standing here in this fashion. So this is ventral, my back is dorsal. Now the way this is named is toward my feet is caudal, and toward my head is cranial. So this is caudal, this is cranial, this is the left side and the right side. And what it's showing is these three spinal nerves. Now remember, we have 31 pairs of these. So we're seeing the segmental nerves coming out of the cord. Now, if we look up here, we see the white matter on the outside and the gray matter on the inside. And if we follow this out, we see these are the dorsal roots. That's in the peripheral nervous system. This is the dorsal horn. We follow the dorsal root. This is now the dorsal or posterior root ganglion. And out here's the spinal nerve. If we come down into the gray matter again, here's the ventral horn. It doesn't show it here, but the lower motor neurons will exit the cord through the ventral root, or the anterior root, and then they will join, those neurons will join the sensory neurons out here in the spinal nerves. And you see this over and over and over again, 31 pairs all along the spinal cord. And a similar pattern in the brainstem, because that's where we find the cranial nerves. And when you take a neuroanatomy course, if you take a neuroanatomy course, one of the things you'll discover is the brainstem is just a kind of highly elaborated spinal cord. All the structures we see segmentally in the spinal cord are more or less repeated in the brainstem. It's just they're highly elaborated. They've had a longer time to evolve. And if you look at this whole system, this dates way, way back until we were segmented animals. Didn't really have a brain, just had ganglia. And we see this pattern of these segmented structures. And that extends all the way into the brainstem, which is truly part of the brain. So that's the structure of the spinal cord. And this next image is to show the tendon tap reflex. So again, there's the lower limb and the upper limb. We can see here the patella, the patellar tendon attaching to the tibia and attaching at this end to the quadriceps muscle. This is a nice figure because what it does is it summarizes everything I drew on the board for the myotatic reflex yesterday. But one of the things I want to emphasize and hold on to as we walk through this is that this is a really, really unusual synapse. This is a really, really unusual reflex because the primary sensory afferent is going to synapse directly under the lower motor neuron. And more commonly what we see is there's an interneuron that we showed yesterday. But this is sort of the classic reflex that we commonly show. So this little spring-like structure is meant to represent the stretch receptors embedded in the muscle. When we pull on the muscle, we activate those stretch-activated channels. We generate or produce a generator potential that then initiates an action potential that propagates along the conductal region of the primary sensory afferent. This is meant to show the cell body in the dorsal or posterior ganglion. The process extends into the spinal cord. In this reflex, at number 4, the primary sensory afferent synapses onto this green lower motor neuron. The lower motor neuron is located in the ventral horn. It's in the gray matter of the spinal cord. It sends a process out into the ventral root, so now it's in the periphery. This then joins the spinal nerve and finds its way, during development, will find its way to the quadriceps muscle. So that's the anatomy. Primary sensory afferent synapses on to lower motor neuron, but functionally what's happening is the action potentials in the primary sensory afferent are releasing the neurotransmitter glutamate onto this lower motor neuron. If we've got enough glutamate released here, EPSPs are big enough, and they almost always are, the action potentials are fired, and they now conduct and propagate to the muscle, where at this site, they'll release the neurotransmitter acetylcholine and cause muscle contraction. That's the myotatic reflex. But there's also the idea of reciprocal inhibition that we discussed yesterday, and the important concept that all inhibition, at least for the muscular system, is in the central nervous system. And that's shown here. Here's the pathway of the primary sensory afferent. Here's a branch now of the very same primary sensory afferent that's now synapsing onto this inhibitory interneuron. This will release glutamate. This synapse will release glutamate just as this one does. We turn on, activate the inhibitory interneuron. It propagates action potentials to its synapse here onto this lower motor neuron. And now we're releasing the inhibitory neurotransmitter. We're in the spinal cord, so it's probably glycine, but it could be GABA. That opens chloride channels. And in so doing now, this is going to inhibit and lower the probability of action potentials being fired here in this lower motor neuron. So that's where inhibition occurs. And you want to do this because you don't want to be activating your extensors and your flexors at the same time. You want primarily extensor activity or primarily flexor activity. And in this way, we inhibit the flexor, the hamstrings, when we activate the quadriceps. And again, the same story here. This lower motor neuron will innervate muscle and release the neurotransmitter acetylcholine. We don't inhibit the release of acetylcholine here. We inhibit the release of the activity of the neuron back here by inhibiting the cell body or input region of that lower motor neuron. So there's a lot in that. So walk your way through that a few times and make sure you see what's going on at each of those steps. Then this next slide is to summarize the electrical activity. So out here, this is what they call a muscle spindle. This is the stretch receptor. The more you pull on it, the larger the generator potential. That means the more of the stretch activated channels are opening. And that now will initiate action potentials that propagate along the conductal region all the way to the output region of the primary sensory afferent. We have a monosynaptic pathway, so this primary sensory afferent is synapsing onto the lower motor neuron. Then fire action potential in the lower motor neuron. This releases acetylcholine onto the muscle, and we activate the muscle. We're going to spend a lot of time, probably tomorrow, on how we activate the muscle and the series of events that occur there. Yes?
Is the primary sensory afferent one cell or a series of cells?
This is a wonderful question. It comes up all the time. Is the primary sensory afferent a single cell or a series of cells? It's one cell. It's one giant cell. And not only is it one cell that does all of this, remember, this is going to be part of proprioception. So it has a branch that will now enter the dorsal columns and ascend the cord. And so these are enormous, enormous cells. So you can imagine, as large as they are, how much protein synthesis activity has to be going on in that cell body. And then the proteins now have to be divided to go either out into the periphery or into the central nervous system. And I don't think anyone knows how that division is made as to where they go. Now, this next slide is beginning to think about more than just the reflex arc. What we've talked about up to now is just the reflex arc. Now what we want to think about is how we're going to deliver information to the central nervous system. How are we going to take information and get it beyond just the spinal cord. I should say deliver it to the brain rather than the central nervous system, because the spinal cord truly is part of the central nervous system. So this is the dorsal ganglion. Here's its neuronal cell body. We can follow it in. We notice that one pathway goes down to the ventral horn, but it will have a branch that now will enter with a whole bunch of other branches of other primary sensory afferents in the dorsal columns. And that's more or less shown in the next slide. And this slide is trying to summarize all of the tracts that we're going to be talking about. These are the tracts in the spinal cord. And again, I remind you that when we talk about a tract, what we're talking about is the grouping, the bundle of conductal processes. And so if we look at this, here's the gray matter. Now everything outside is the white matter. Now they've shown this as regionalizing the white matter into different tracts carrying different information. Another thing to notice is that we're bilaterally symmetrical. What we see on one side, we see on the other. In blue is sensory. And what we talked about yesterday were the axons from the primary sensory afferents that came in and then branched and entered this dorsal column system. The dorsal columns are axons, conductor regions of primary sensory afferents carrying information about discriminative touch and proprioception. And again, they're headed to the dorsal column nuclei. But there's another area over here. This is also sensory. We're only going to look at one part of this. If you take an anatomy course, you'll see there are several different aspects to this so-called ventral area, the ventral spinal. What we're going to talk about is the spinothalamic tract. We're not even going to talk about the corticospinal tract, this ventral one. We're going to talk about the lateral one. But first, this is where we're going to find the spinothalamic tract. These are going to be the axons, conductor regions of second-order neurons in the spinothalamic tract. And we'll walk our way through that in just a minute. In red, we see motor. There's this pathway called the ventral corticospinal tract I'm not going to talk about. And the one we're going to talk about is the lateral corticospinal tract. And it's going to contain the axons, the conductal regions of upper motor neurons located in the motor cortex. And those conductal regions are going to then end with an output region on lower motor neurons in a monosynaptic pathway. But let's finish the slides here for a moment with this is a summary that a good friend of mine, Dr. Anna Fales, drew some years ago. And I really like it a lot because it's so simple. If you know what's going on in this figure, you really understand the dorsal column or posterior column medial lemniscal pathway. She's showing the neuronal cell body here. She's showing the input region here. It enters the cord. One branch goes into the ventral horn. The other branch now ascends the cord in that dorsal column system. It has its first synapse here, the primary sensory afferent, synapses onto the second order in the dorsal column nuclei located in the brainstem. That's what we drew yesterday. Remember, primary sensory afferent never crosses the midline, but the second order neuron always does. So the primary sensory afferent synapses onto the second order neuron, it crosses the midline, and now it continues to ascend the cord until it forms a synapse in the thalamic relay nucleus. So now we have a synapse between the second order neuron and this third order neuron, the synapse in the thalamic relay nucleus, and now the third order neuron sends a process and now synapses onto neurons in the somatosensory cortex. Again, when we talk about the cortex, we'll see that that's always organized into 6 neuronal cell body layers. And in this particular case, these are now going to synapse onto the neurons in layer 4. Don't worry about that detail. Just recognize that this third order neuron is sending a process up to synapse onto neurons in the motor cortex. And from there, that information is going to radiate up to other parts of the brain so we make a decision about what's going on. Yes?
Is the medial lemniscus the same thing as the dorsal column nuclei?
So what's the difference between the dorsal column nuclei, and the medial lemniscus? The dorsal columns, these are the dorsal columns. If you go back to, let me go back. These are the dorsal columns. On this side of the midline, they're carrying information from the same side. This is the dorsal columns on the other side. Now, the dorsal columns are nothing more than axons. They're nothing more than conductal regions. So they're an extension of those sensory neurons. So then, again, the neuron goes all the way up the periphery, comes into the brain, makes that synapse in the ventral horn, and then another branch of the very same neuron enters the dorsal column system and ascends the cord in the dorsal column system until it reaches the brainstem. At that point in development, it forms a synapse between the primary sensory afferent, the output region of the primary sensory afferent, and the second order neurons. And the second order neurons are now, again, their cell bodies are on the same side of the body as the primary sensory afferent's cell body. But now that neuron, which receives input from the primary sensory afferent, is going to cross over the midline, and it's going to do that in the brainstem, and that's the medial lemniscus. Okay? So the dorsal columns are the axons or conductal regions of the primary sensory afferents. Then it forms a synapse onto the second order neuron. And in this particular pathway, that bundle of second order neurons has a name, and that's the medial lemniscus. That's the bundle of axons in that tract. You could talk about it as being the next one. You could talk about this as being they probably call it bulbo because it's in the spinal, it's in the brainstem. This could be the bulbothalamic tract. But it's the same thing as the medial lemniscus. It's just the medial lemniscus gets a special name in this pathway, which is the second order neurons. Second order neurons go to the thalamus and synapse onto third order neurons in the thalamic relay nucleus. But again, it's 1, 2, 3. 1, primary sensory afferent. 2 is second order, crosses over, synapses into third order in thalamic relay nucleus. And that's the pattern for all somatosensory systems.
Am I right in thinking that it's the cell body and the dorsal column nuclei and the axon is the medial lemniscus?
The second order neuron input region or cell body will be found in the dorsal column nuclei. The processes will be in the medial lemniscus. Now, if you wanted to include the input regions of the medial lemniscus, I think you could do that. But that's just a question of consistency. Where are we going to? But the medial lemniscus is clearly the part that crosses over and contains those axons. Anything else? Okay. Then let me go on and now what we're going to do is we're going to draw something known as the spinothalamic tract. The spinothalamic tract is going to carry information about pain, pressure, and temperature. So let's do exactly what we did yesterday. We're going to start out with a cross-section. We'll delineate the white matter and the gray matter. We'll extend the dorsal horn out into the periphery as the dorsal root. We'll put a neuronal cell body in here and extend the process out in both directions from the neuronal cell body. This again is the cell body of the primary sensory afferent. We can imagine, I'll take this out here, and we can imagine in this case, put it this way. We can imagine this neuron originating, having its input region out in, say, your big toe. So if we follow it out, it's your input in your big toe. And we're going to say that this is a pain neuron. We could equally say it's a neuron sensitive to temperature, cold or heat, or a neuron sensitive to pressure. But in all those cases, just like we talked about a moment ago with the PCML, the neuronal cell body is in the dorsal ganglion. It's a single neuron with a process that goes from all the way from your toe all the way to the dorsal ganglion in the neighborhood of your lumbar spinal cord, someplace in here. The neuron then sends a process into the central nervous system. And you remember we made a big deal just a moment ago about how that would stay on the same side of the cord. It does. But almost immediately after entering the dorsal horn, it synapses onto a second order neuron. So here's the neuronal cell body. This is the input region of the second order neuron. Remember the rules. First order neuron, primary sensory afferent, never crosses the midline, but the second order neuron does. Now, as we just discussed, the second order neuron for the DCML crosses over in the medial lemniscus. But we don't do that here. What we do is we take this second order neuron and we send it over to the opposite side of the cord, and it enters a region over here in the white matter, the other side of the cord, a region known as the spinothalamic tract. Again, there's meaning in the nomenclature. The tract tells you it's a bundle of conductal regions. Spinothalamic tells you its neuronal cell bodies in the spinal cord. Its output region is going to be in the thalamus. So that means that this green neuron is going to enter the spinothalamic tract, and it's going to ascend the cord. And it tells you it's going to go all the way to the thalamus. So let's take this same image and imagine it, rather than in cross-section, we imagine it now in our ice cream cone diagram. Let me make that a little farther down. Where this is the brainstem. So we put a dotted line across here. That would be the interface between the brainstem and the spinal cord. Just an anatomical landmark. Then we're going to imagine that we're someplace down here in the lumbar region. And if we repeat the drawing we have here, here's our crude foot. This neuron, whose cell body resides in the dorsal root ganglion, this neuron has a process that goes out to your toe. But now we'll follow it in the central nervous system. It comes into the central nervous system. Almost immediately after entering this central nervous system, as we show here, after entering the central nervous system in the dorsal horn, we label this as the dorsal horn, it synapses onto that second order neuron. Now remind you that we're bilaterally symmetrical and we have a kind of midline, and that the second order neuron crosses over the midline and enters this spinothalamic tract. It enters that tract, and now that tract now ascends the cord. This will be one neuron with its cell body in the dorsal horn, ascending the cord all the way through the brainstem, ending up here in part of the midbrain, known as the thalamic relay nucleus. So again, it's the spinal thalamic tract. It has the cell body in the dorsal horn, has its output region in the thalamus. And you'll remember from yesterday that the third order neuron now extends from the thalamus and has its output region in the somatosensory cortex of the cerebrum. So you see the pattern's the same, 1, 2, 3. 1 never crosses over, 2 always crosses over, forms synapses in the thalamus, thalamic neurons then synapse onto neurons in the somatosensory cortex. So let's now, so any questions up to here? Those are the 2 pathways, yes?
So this is for pain in general?
For pain, 2 other things. I'm going to make Sam. You opened yourself up.
I don't remember.
Okay, one's going to be pain, one's going to be temperature, so heat, cold, and third's going to be pressure. And that's distinct from the fine touch and proprioception that we saw in the dorsal column system. Yeah?
I think there was a question on the lecture quizzes yesterday about the synapse of the third order neuron, if it was in the parietal lobe or the frontal lobe. I don't know if that was specific to.
Yes, we're going to come to that.
Okay.
We're going to see that the synapses here between the third order neuron and the neurons in the cortex, that's all going to occur on something known as the post-central gyrus. And the post-central gyrus is a part of the brain, I'll show you here in a minute, that sits right behind the central sulcus. Let me show it to you. I may as well. So this is an attempt to give you some sense of what the brain looks like. And this is the brain stem here. This is the cerebellum. We're not going to talk about it. The diencephalon or midbrain is buried underneath this. This is the structure of the cerebrum. This is a big part of your brain. We like to think it's the most important part of our brain, but the more we delve into it, we find there are other parts that are doing things we don't think about, but they work just fine. But what we see is we can divide these regions anatomically. So we can divide the regions into this region up here, which is beneath the frontal bone of the skull. This is the frontal lobe. There's a region here called the temporal lobe because it's beneath the temporal bone of the skull. There's a region back here called the occipital lobe, which is part of, it's sort of the back of the brain, and it's underneath the occipital bone. And up here is one under the parietal bone, and this is the parietal region. What we look at on one side, we look at on the other. We're bilaterally symmetrical. And the other point this figure makes, I think, nicely is that what they've done here is they've cut away, in this imagined view, they've cut away a bit of the cerebrum showing the frontal lobe and the frontal lobe and the temporal lobe. And what they're trying to show here in brown is that the cortex is a very, very thin layer. It's a veneer over the top. And if they would show more detail, they would show that there are 6 neuronal cell body layers in that cortex. Wherever you look, it doesn't matter whether you're looking up here in the frontal lobe or back here in the occipital lobe, they're always organized into 6 layers. But what we're going to be talking about today are 2 parts. And I'll show you this as we come back to it. But the 2 parts, and the question is, when you look at the anatomy, are there landmarks you can recognize? Absolutely. If we would take the skull of anyone in this room and open it up and look at the brain, we'd see the same landmarks. They're all identical. And one of those major landmarks is this division between the frontal lobe and the parietal lobe. And this is known as the central sulcus, sometimes, because it's so big, known as the central fissure. Now, we're going to have different neurons in these 2 places. We have different neurons all over the brain, but we'll organize them into functional regions. And the functional regions we're going to talk about, and this pertains to the question, is that we're going to talk about the region here, which is on the postcentral gyrus. If we would look at the cortical layer right beneath the surface on the postcentral gyrus, that's somatosensory cortex. If we hop over the central sulcus and look at this region, that's the precentral gyrus. If we look at the organization of neurons in the cortex of the precentral gyrus, that's going to be motor cortex. So again, an idea that we're going to separate function in the brain, in this case, an extreme case, where one's motor and one's sensory. If you're to get into this, you'll find that much of vision is down here in the occipital cortices. Much of hearing and language is done here in the temporal lobe, or temporal cortices. So just the idea that different parts of the brain are doing different things, and those different things are relegated to different cortices, but all of these cortices now are going to be connected one to another through the white matter, which resides below the cortex. Okay? Yes?
So the DCML pathway synapses in the frontal lobe and the spinal thalamic, because it's...
Almost, not quite. What we're going to see is that the output regions of the third order neurons for the somatosensory system, whether it's the DCML or the spinal thalamic tract, they're going to have their outputs here on the postcentral gyrus. The upper motor neurons for the motor system are going to be found in the cortex of the precentral gyrus. So again, precentral gyrus is motor, postcentral gyrus is sensory. And I'll show you that in more detail. But again, this is the simple diagram of the dorsal of the spinal thalamic tract. Primary sensory afferent synapses on to the second order neuron and the dorsal horn, crosses over, ascends the cord now to the thalamic relay nucleus. This tract is the spinothalamic tract. Third order neuron goes to the somatosensory cortex. 1, 2, 3, the rules are the same. The only real difference between those tracts and the information they're carrying is where the second order neuron crosses over. So let's take that information and I'm going to redraw this, and we're going to talk about something known as Brown-Séquard syndrome. And I want to walk through this for a couple of reasons. First, that Brown-Séquard syndrome is a particular syndrome. It in fact occurs. If there's been an accident and the spinal cord is cut right to the midline, there's certain signs that these individuals will express. And it's a first indication here of how, by understanding the neuroanatomy, by understanding where neurons originate, where they send their processes, where they have their terminals, by understanding that neuroanatomy, when there's a lesion in that system, you can understand where the lesion is. It's one of the reasons that most most neurologists are outstanding neuroanatomists, because they're trying to suss out where the lesion is in the nervous system from what's broken. And they have to understand the anatomy to know where that lesion happens to be. So this is an example of that. So let's start. And we're going to imagine again the ice cream cone diagram. Our spinal cord. We're going to work down in the lumbar region. As we did above, but you could do it on any one of these segmental nerves. And we're going to see again that we have neuronal cell bodies. Let me make these a little bigger so we have a little more room. What I'm trying to do with these brackets is make the dorsal or posterior root ganglia. So this is a neuron, goes out, we'll draw the leg again. That neuron will have innervate the quadriceps muscle, because it's going to be carrying information about proprioception. It could be from the skin carrying fine touch. Put the muscle in here. And the other neuron, whose cell body resides here, one we just talked about, we're going to talk about it having its origins down in your toe. It's a pain neuron. It could be a pressure neuron, could be a temperature neuron. It has its cell body in the dorsal root ganglion, but we're bilaterally symmetrical, so we can draw the same thing on the other side. Here's the quadriceps muscle. Now remember, when we talk about this, we're talking about one neuron, but there'll be hundreds of these neurons in the dorsal root ganglion. So here's one, has its input region here in the muscle. The second one is pain neuron coming from your toe. Now let's follow the processes, just like we did before. This is a primary sensory afferent coming from the muscles carrying proprioception or fine touch. It ascends the cord on the same side, forms synapses up here in the dorsal column nucleus. Now, I'm just going to show it for one of these. Then there's the medial lemniscus that crosses over and ascends the cord to the thalamus, where it synapses on to a third order neuron. This is going to get busy enough, so I'm not going to draw in all of these for all of those sites. So then let's follow the pain neuron. Comes in, forms a synapse. Second order neuron crosses the midline. Here's the midline. Second order neuron crosses the midline, ascends the cord, all the way up to the thalamic relay nucleus. So that accounts for the sensory input from the right side. Now let's look at the sensory input from the left side. So say this is right, this is left. The primary sensory afferent carrying information about proprioception and fine touch enters the cord, ascends the cord in the dorsal columns. Will now form synapses on the left side, the same side it entered, in the dorsal column nucleus. Synapses onto the second order neuron crosses over the midline and ascends to the thalamic relay nucleus. So now let's follow the pain neuron. Pain neuron comes in, synapses onto the second order neuron in the dorsal horn. The axon enters the spinothalamic tract, ascends the cord on this side, all the way to the thalamic relay nucleus. Now let's define Brown-Séquard and think about what's going to happen here. Brown-Séquard is a hemisection of the spinal cord. Hemi means half, so it's a section to the midline. So we can imagine here that something terrible has happened. There's been a car accident, a gunshot wound, a knife wound, and we have cut the spinal cord right to the midline. So now let's ask the question, what can this person who's undergone this dreadful accident, what can they still feel and what can't they feel? What's broken? We've broken the wires here. The wires headed to the brain are broken. So they're no longer carrying information. So what information gets through and what information doesn't? If you can follow this, you'll understand these pathways. Okay, so let's look at this side. We pinch the toe. Pinch the toe hard enough to cause pain. Does this person feel that or not? Well, they'll feel it, right. So it crosses over, it's not interrupted, so it's working fine. But now we ask the question, about proprioception or fine touch. Usually this will be fine touch. So they'll lightly touch the skin or have these calipers which show 2-point discrimination. And they'll ask the question, can you feel that? Well, you can see it's busted right here. So this information is not getting through. So from the right side, this person has no sensation of proprioception in the lower limb, nor do they have sensation of fine or discriminative touch. But let's go to the other side. Here on the left side, if we pinch this person's toe, will they feel it? No, because it's broken right here. But the proprioception and fine touch gets through just fine. And these are the kinds of things the neurologists do all the time. They puzzle out what information is getting through, what information is broken, and knowing all of these different pathways, having a detailed understanding of all the different pathways, they can typically suss out where the damage is. But it takes good neuroanatomy to be able to do that. And neurologists are typically great neuroanatomists. So that's the basic pattern. Now, it's time. Let's go ahead and take a break. And what we'll do next is we'll deliver this information from the thalamus to the somatosensory cortex. And we'll talk a little bit about what's going on in the somatosensory cortex and the concept of somatotopy. Okay, so what we want to do now is we want to think about the final inch, as it were. We want to think about how those neurons, whose cell bodies are in the thalamus, are going to distribute themselves and their innervation pattern in the somatosensory cortex. So I'm just going to take an example here. I'll go up to the upper panel, and I'm going to enlarge what I have on the left. So this is meant to represent the somatosensory cortex. What we see on the left, we'll see on the right. Now, it's a detail, and I'll never ask you about this, but it turns out that wherever you look in the cortices, there are 6 layers of neuronal cell bodies. And the third-order neurons, which we typically show in red, where they're coming from the thalamic relay nucleus. These neuronal cell bodies now extend from the thalamus and find their way to layer 4. Of the somatosensory cortex. Did I say that right? Is it 4 or 5? 4, okay. So it goes to layer 4. And now this is going to be an excitatory neurotransmitter. It's going to be releasing glutamate onto neurons in layer 4. And it doesn't matter whether this is a neuron carrying information about from the DCML, or whether it's carrying information from the spinothalamic tract. You'd have another one over here that may be carrying information in the spinothalamic tract. It's also going to go up here to layer 4. And we'll see the same thing in the other side. Now, what you have to imagine here is that we've only talked about 2 neurons. If we follow this, we followed the 2 neurons that came from the lower limb. There'll be thousands and thousands and thousands of these. And they're all going to be feeding into the thalamus. They're now going to be feeding into the somatosensory cortex. And the idea that I want to emphasize here, I'll erase this, and then this is a concept. It's a concept in neurobiology known as somatotopy. And the definition of somatotopy is the map of the body plan on the cortex, or on the cells of the cortex. So we'll spell that out and say that somatotopy is defined as the map of the body plan onto neurons. In the cortex. In this particular case, it's going to be the somatosensory cortex. And when we look in the brain, we find the somatosensory cortex is located on the post-central gyrus. This was worked out in animals fairly long ago, but this wasn't worked out in humans until the late 1940s, early 1950s. And the way it was worked out was by a surgeon. And the surgeon was Wilder Penfield. And Wilder Penfield was world-renowned as being one of the best brain surgeons ever. And at this time, there were not very good drugs to control epilepsy. And epilepsy can be a debilitating neurological disease. You get these seizures every so often. And he recognized that if he could go into the brain and find the site where those seizures were originating, if he could remove that site, sometimes known as the kindling point, if he could remove that site, he could then relieve much of the seizuring. But he also knew that if he went into these brains and started willy-nilly pulling out parts of the brain, he was going to have effects on these individuals and could change their behavior for the rest of their lives. So it was a delicate sort of thing. So one of the first things he needed to know was what kind of information are we seeing in various parts of the brain. And the story is, first, I think it got more sophisticated, but the story is when he first did this, he had people awake. And you have no sensory information coming from your brain per se, so you don't feel that. But they would inject local anesthetic into the scalp, cut the scalp, pull the scalp back, then take a bone saw and cut the skull cap out. Well, this person's wide awake. It's astonishing. Pull off the skull cap, and then he would literally take his finger and start poking around. And when he poked in this particular region that was on the post-central gyrus, depending on where he poked, people would say, oh, I feel that in my hand, or oh, I feel that in my foot, or oh, I feel that in my face. And he found that from individual to individual to individual, these areas were similar. And what's more is he found that particular regions of the somatosensory cortex could predictably be seen to activate sensory neurons, be responsive to sensory neurons from a particular region of the body, say the face or the hand or the foot. And it was always in the same spot. And that led to this idea of somatotopy and the map of the body plan on the surface of the brain, on the cortex. Let me show you a couple of pictures of that. So what we're seeing here so now to orient you. This is the image of the brain, much like we just saw a moment ago. And it's as though I'm facing this wall, and you're looking at the left side of my brain. What you see on the left side, you'll see on the right. And there's this demarcation point between the frontal lobe and the parietal lobe known as the central sulcus. It's a clear division. We'd see it in everyone in this room, anywhere we look. It turns out that when Wilder Penfield was poking around, he found that when he would poke on the post-central gyrus, it would all be sensory. When we would poke on the pre-central gyrus, he would get movement, actually get people to move a hand or a foot or what have you, depending on where he poked. This is the first indication of the way the body plan is mapped out on the somatosensory cortex. And this is a kind of demonstration of that. Every time we would go in, we would find that the feet are represented on this region of the somatosensory cortex. The body or trunk is located here, the hands are up here, the face is here, the jaw and the tongue. Now, one of the things I want you to notice is that it's not consistent with the anatomic size of the individual region. It's really exaggerated. The face is way bigger than the foot. The hand is half as big as the whole face, and is much, much bigger than the rest of the head or the trunk. And the reason for that is that we are very tactile creatures. We have to do a lot with our hands. I mean, if you're a dog or a cat, you don't have to worry about playing the piano. But we do these very delicate things with our fingers. And so we have a lot of sensory input coming in, telling us where our fingers are in space. We have a lot of sensory input telling us how to feel things, because we get along in the world largely by proprioception. So we have a tremendous area of the somatosensory cortex that's dedicated to the hands. We also have a large area that's dedicated to the face. And that's probably related to what I'll show you in a little bit, the motor cortex. We need to know in space where our face is. We need to know where the muscles are. We need to know how they're being expressed, because much of our interactions as primates comes through muscles of facial expression. So we need sensory information coming in telling us what our muscles are doing so we can express one feeling or another, whether it's joy or sadness or anger, through the muscles of facial expression. So we need to know where all of those things are. Moreover, as humans, we have language. We make sounds. And those sounds now are regulated by things like the lips, and they're regulated by the teeth and by the tongue. So we have a large part of the somatosensory system dedicated to knowing where our tongue is in space, so that we can place our tongue in the right spot to make consonants, to use the larynx to make vowels. And all of that's done by this enormous amount of sensory input. If you were to look at this in an elephant, you would see it in a tremendous area taken up by the trunk. Because if you look at the way that an elephant interacts with the world, it interacts with its trunk. Or a horse, it would be its lips. If you do this in a mouse or a rat, you have a tremendous area of the somatosensory cortex taken up by the innervation from the whiskers, the vibrissae. Because mice and rats in the dark interact with their environment by feeling around with their whiskers. So they have a tremendous area of the somatosensory cortex dedicated to what's coming in from the whiskers. And it's all that. If you look at different animals, they have different areas. You had a question?
If somebody had like a speech impediment, was that more like cultural, or would you find that like,
Ooh, that's a hard question. The question is, if there are speech impediments, is it in the wiring or is it cultural? My guess is there's some of each, but I suspect that most of it is cultural. Some of it may be how you hear. I mean, what we're doing as we form our ability to make language is we hear language and then we learn to make those sounds. There's some magical connection here that I don't understand between your auditory system and this motor system. And there are these great videos. There's a language, I think it was an Inuit language, where there are sounds in that Inuit language that the rest of us don't make. So it's a particular sound, it's unique to that language. And if you look at a child, a baby, before they've acquired language, they will respond to that sound. But if you look at someone who speaks another Western or even Eastern language where that sound doesn't exist, those individuals can't hear it. They don't respond to it. So there is some wiring going on, but the wiring is associated in this case with what the child has heard as language. And it's also this issue, some people are better at than others, clearly. But for the most part, rule of thumb is that if you don't acquire your language before you're about 9 or 10, you'll never speak that new language without an accent. Someone who's a native speaker will pick up the nuances of sound. So that's something to do with wiring too, but that's acquired wiring. So it's acquired in terms of what we hear as typical speech, how we hear dialects and accents. And it's also how we rewire what we hear to how we make sounds. And if you don't get that wired up early on, you never get it quite right. You can still speak, you can make yourself understood. I'm terrible with languages, but certainly I know a lot of people who don't learn language until later on, and they get quite fluent and very subtle hint of dialect or accent. So where that is, I don't know, but it's probably someplace in this combination of sensory input and motor output. But it's a great question, and I'm sure speech therapists have thought a lot about this. Anything else? Let me see if I can find the figure that I want to look at. Here it is. This is a three-dimensional homunculus. This is a three-dimensional image of what we think the sensory input to the somatosensory cortex would look like in 3 dimensions. And I like to show it because it really exaggerates and emphasizes everything on that last slide. I mean, look at the representation of the hands. Look at the representation of the tongue and the lips. I mean, Mick Jagger doesn't have lips like that. That joke is aging with me and Mick Jagger, but you see how exaggerated all of that is compared to the rest of the body. And that's simply that we have this map of the body plan on the surface of the cortex. Any questions on that? Well, if not, let's now, I'll do this in a drawing and then I'll come back to some slides. Let's now switch from the somatotopy of the sensory system and now move over to the motor cortex. Now, where the sensory cortex is located on the post-central gyrus, we're going to see that the motor cortex is part of the frontal cortex and is located on the pre-central gyrus. But again, just as we saw somatotopy for the sensory systems, we again see somatotopy for the motor systems. But let's walk through what these motor systems are going to look like. And as we do this, any of you who are interested in veterinary medicine will probably never see this again, because this is almost entirely unique to primates. And one of its hallmarks is it's a monosynaptic pathway between the upper motor neuron found in the precentral gyrus in the motor cortex and a synapse onto the lower motor neuron in the spinal cord, single synapse. In most quadrupeds, you don't see this. Most of their systems will go from something called the red nucleus in the brainstem and then activate muscles. But because we're so unusual, this ability to very carefully regulate movements of the hand and movements of the face, we have elaborated the system. So we're going to talk about the descending motor pathways. Again, this is sensory in, we talked about a moment ago. Now this is motor out. I hope I get some time here. I'll tell you a little bit about the association cortices where we'll integrate this information. But let's start with the same idea we had before. This is meant to be the cortex. Let me put that in a different color so that it's just more of a contrast from what we just talked about, because we talked about putting sensory in blue. Let's go ahead and put the motor in green. So we're going to label these two ovals as the motor cortex on the pre-central gyrus. This is part of the frontal cortex. As before, as always, there are 6 layers of neurons. And here the main player is going to be in layer 5. And this neuron has a number of names. We're going to call it the upper motor neuron. Sometimes it's called the Betz cell. Betz was I think he was a Ukrainian physiologist from the 19th century. And they're unusual. They're among the biggest neurons anywhere in the body. And they get their own name called Betz cells. And these upper motor neurons, which are typical pyramidal neurons, will send a process out from the motor cortex out into a region that I'm going to label here as the internal capsule. I'm cheating a bit. And the reason I'm cheating a bit here is that if you take a neuroanatomy course, you'll find that the internal capsule also contains the ascending neurons, the third order neurons that are going to the somatosensory cortex. In the early days when they named it the internal capsule, they bundled them all together. We now know they're different neurons. But as part of the internal capsule will be these axons, conductal regions of upper motor neurons that enter the internal capsule, and they will remain as a single bundle. And what we see on the right, we see on the left. And then, let me put the drawing over here. Back to the ice cream cone diagram. Here's the thalamus in red. And in green, we start out with the motor cortices. The upper motor neuron sends its process out into the internal capsule. The internal capsule, these axons will skirt around the thalamus. They don't go through the thalamus, they go around the thalamus. And they come down into the brainstem. And just at the point where the brainstem is about ready to fuse with the spinal cord, right above your first cervical vertebrae, right in something called the medulla oblongata, this neuron will cross over the midline. So again, a reminder, we're bilaterally symmetrical. And if we follow one of these upper motor neurons from the right side and the left side, we see that the ones from the right side again come down and cross over in the brainstem just before the brainstem fuses with the spinal cord. This region has a name. This is called the decussation of the pyramids. So just as we saw with the sensory system, everything crosses over. That is, in the sensory system, if I would poke you in the somatosensory cortex on the left side, you would feel it on the right side. If I poked you the somatosensory cortex on your right side, you'd feel it on the left side, because all of those second-order neurons crossing over. That's why you have this arrangement of the neurons in your brain are on one side, you recognize them from the other. The same is true here. We're going to cross over. It's just a developmental phenomenon. This happened 1 billion years or more ago, and we're stuck with it. And so the upper motor neuron crosses over, and then it enters the spinal cord. I should say, too, this decussation of the pyramids, this weird name, apparently it arises from the 19th century histologists when they started looking at this part of the brain and they were staining for axons. They commonly saw it in sections that would look like a triangle. And then as they began to run sections through it, that triangle then began to look more and more like a pyramid. And in 3 dimensions, it looked like a pyramid. So they called it the decussation of the pyramids. The decussation of the pyramids is, in fact, the axons or conductal regions of the upper motor neurons crossing over. Decussation just comes out of the Latin. It means to cross over. So that means now those neurons that whose cell bodies originate on the left side are on the right side of the cord. Those who are on the right side are now on the left side of the cord. And they're in a region of the cord, has a name, and there's meaning to it. It's called the lateral corticospinal tract. So let's imagine we'd cut a section through here and look at the spinal cord in cross-section. Now, it's lateral, so that means it's going to be on the edges. It's corticospinal, so that tells you the neuronal cell bodies of the upper motor neurons are in the cortex. Specifically, in this case, they're in the motor cortex. They're located in the motor cortex, which is found on the precentral gyrus. And if we'd look in here, we'd see these little dots as axons. Thousands and thousands and thousands of them. And they would be organized according to the place they originated in the course, in the brains, where they originated in the motor cortex. So those neurons that were going to innervate the muscles of the hand would be in a similar place, and those motor neurons that are going to innervate the muscles of the lower limb, for example, would be in another place. And they will reside similarly all along that tract. But at some point during development, this neuron will get off. A particular neuron will get off and now enter the ventral horn. And in the ventral horn, this upper motor neuron forms a synapse onto the lower motor neuron. So again, this is a monosynaptic pathway. It's a monosynaptic pathway that connects directly from the upper motor neuron to the lower motor neuron. And this lower motor neuron now, as we've seen before, is going to go out and innervate muscle. So that's the overall pattern. Yes, yes.
So to clarify, when you're looking at ascending pathways, you have the 3 order neurons, 1, 2, and 3.
That's sensory.
Yes? Yeah. And then when you're descending, you just have your upper and lower motor neurons?
Okay, so your question is, are you telling me that on the sensory side it takes 3 neurons to reach the cortex, but to go from the cortex to the muscle, it only takes 2? Yes, absolutely. In this pathway, that's true. Yes. So let's have a pic. There's a pretty picture of this. So fundamentally, what we've drawn here is this image. Let's follow the one on this side first. This is basically what we've drawn. Here's the cell body of the upper motor neuron. It enters the internal capsule. This neuron, it's one axon, one extremely long axon, well then can be followed from the midbrain to the pons to the medulla. And now once we get just to the point where the medulla joins the spinal cord, it crosses over in the decussation of the pyramids. Goes to the other side of the brainstem, enters the spinal cord, and now remains in the lateral corticospinal tract until such time it signals during development to leave the lateral corticospinal tract, enter the ventral horn, and in this case innervate a lower motor neuron that innervates the muscle of the thumb. Now you notice this is the left hand, this is the right side of the brain. That's because of the crossing over and the decussation of the pyramids, but we could follow the same thing from the other side. This image is showing the representation on the motor cortex. And again, it's the body plan mapped out. It's somatotopy. And again, it's not equivalent to the anatomic size. It's equivalent to the number of upper motor neurons we have for each of those regions, and it tells you something about the degree of innervation of different parts of the body by these upper motor neurons. And again, you see an enormous area innervated by the hands. That's so we could make stone tools a long time ago. Now we do things like play the cello. But we have tremendous dexterity in our hands. We can do things that, and we have these, what they call fractionate movement of our fingers. And that's because of this direct connection between the upper motor neuron and the lower motor neuron. But the other place where you see an enormous area represented is in the face. And that's because we have in our face these muscles of facial expression. They're innervated by a particular, particular cranial nerve 7, sometimes known as the facial nerve. But these are important to us because when we look at someone else, we're trying to figure out what they're thinking. And the way we begin to figure out what they're thinking is by their muscles of facial expression. We do a lot of expressing what we're thinking through these muscles of facial expression. And to me, it's an interesting story, and I'm going to exaggerate this a bit. But as people age, one of the things they do so they don't look quite so old is they'll go to a dermatologist, and they will have a dermatologist inject botulinum toxin around their face, in the muscles of their face. And what that does is that botulinum toxin goes into the axon terminals of the lower motor neurons and destroys the vamp. It destroys that molecule that's important for synaptic vesicle fusion. So you lose the ability to control those muscles. And the irony of this to me is that some of the people who use this a lot are actors because they want to look younger. It's just the drive in that industry to have people look younger, so they go get Botox injections. But to me, the irony of it all is that then as their profession dictates, they go on set and the director says, show me anger or show me worry. And they say, I no longer have control of those muscles. Now, I think that's an exaggeration. I think they still do. But it's still an irony to me that in order to look younger, we'll sacrifice this ability to have the fine control of muscles on our face, which are really important in our communication. But again, that's all comes from this idea of the enormous area that's dedicated to muscles innervating the face. And some of that too is lips and tongue. And that's language. We need to be able to know where our tongue is in space. We need to be able to, in short order, place the tongue in a particular spot to make a particular sound. It's remarkable how fast we can move our tongues around to make those different sounds. Stop consonants with the tongue against the roof of the mouth. S's where we open things up and hiss the S out through the teeth. All of that's done by having the right position of your tongue and your lips and your jaw at the right moment. And we switch them every fraction of a second to make different sounds. It's astonishing. And all of that's done by this very highly ordered motor system. Well, last bit. I promised you that when we talked about this, I would try to put the 3 boxes together. Let's see if I can do this here. Where did I put it? Here it is. This is the wrong one. This is actually something that came again from Dr. Fales when she teaches her anatomy course. And I'll try to go through this fairly quickly. Right here, that's the central sulcus. So from what was just said, you'll know this is somatosensory cortex. You'll know that this is motor cortex. So the motor cortex, in large measure, is going to be the final decision point of what muscles you're going to activate through that descending pathway. But we said at the very beginning that we had a 3-box model: sensory in, integration, motor out. So can we think a little bit here in a very superficial way about the integration? And in fact, what's happening is there's a region back here. This is going to be in the parietal lobe. This is going to be the parietal cortex. And this is a region is known as the parietal association cortex. And the parietal association cortex is taking information in from all these different sensory modalities. So as you're approaching the world, you're getting information about where your body is in space. That's all somatosensory. You're getting information coming into that area from vision. So we have vision coming in from the occipital lobe. You have information coming in from sound from the temporal lobe. All this information is coming into the parietal cortex, and it's now at that site being integrated in ways that we don't quite understand. But we're now integrating the information of all the world around us. If you just have the somatosensory cortex, you don't get much information about the world. You really have to have it integrated with everything else. That's going on in the parietal association areas. And in the parietal association area, then, we'll take this information, and I'm going to simplify it a lot. It will take this information and send it up to the forebrain, up to the frontal lobes. And there are cortices in the frontal lobe, particularly in here, which are called the premotor areas. The premotor areas, it appears, that's where you make a plan. And there's some data, I'm not sure how much stock to put in it, but there's some data that you can take recordings. And you can take a skilled basketball player, for example, it's one that's commonly used, and say, imagine shooting a foul shot. And athletes do this all the time. They picture what's going to happen. And that gets set up as a series of patterns in this premotor area. So when the basketball player is going to shoot a foul shot, they're picturing standing on the foul line. They're picturing the distance to the basket, and they're imagining what motor neurons they're going to activate to put the ball through the hoop. And that's all planned out in the premotor cortex, and we think then that information is released as a kind of cassettes of activity that go to the motor cortex to release that information. I'll tell you the story, it may make some sense or not. Years ago, my son was a competitive fencer. And I used to watch how the coaches would teach him how to fence. And one of the things that you do in fencing is called a feint deceive. What you do is you know what you're going to do. So you make a movement that's not the movement you're intended, but you start to make a movement, and your opponent has seen this 1,000 times. And so the opponent then immediately thinks they know what you're doing, but that's the feint. That's deceptive. And the opponent makes this movement that's inappropriate, and you follow in and get the touch. And that's all in this system, because someone sees something, and before they can realize what they're actually seeing and interpret it, it's too late. They don't know what motor output to do. But the person who's making the move, they've set this all up in the premotor area. They release a cassette of activity to the motor cortex, and off it goes. And so there's a tremendous amount of planning that goes on here. Another example here that I find astonishing is if you imagine a batter in baseball. The amount of information that's coming in at any given time is amazing. They have a pitcher standing 60 feet 6 inches away. The pitcher is going to release that ball, and it's going to be on you in 0.6 of a second. In that time, you have to compute the spin of the ball, the position of the ball, and where you think the ball is going to be. Hitters say they watch for the release point. Some hitters say they can see the spin. I don't think they consciously determine that. I think all that information comes in through the visual cortex and somatosensory cortex, so you know where your body is in space. That information is integrated in the parietal association areas, and then that sends out information to the motor cortex and the premotor cortex to say, put the bat in the right spot, swing it at the right time, and make contact with the ball if things go well. All of that happens in a fraction of a second. And we have these incredible machines that allow us to do this. Now, it takes practice. You don't do that the first time. It takes years and years of practice. And some of that practice is in terms of seeing it. Some of that practice is in terms of knowing what motor cassettes to set up, and then executing those motor cassettes so you carry out the right motion. It's an amazing machine. And so that's the final piece here I'd like to finish with, which is this idea of integration and output. And what we'll do tomorrow is we'll follow the lower motor neuron out from the central nervous system, out through the ventral root, out to muscle. And we'll ask the question, how does that lower motor neuron activate the muscle? We've seen some of this. And we'll see the lower motor neuron releases neurotransmitter, binds to a ligand-gated receptor. Ligand-gated receptor generates an end plate potential, or an excitatory postsynaptic potential, that if it's big enough, and it almost always is, fires an action potential in the muscle that leads to contraction. And that will be most of what we'll talk about tomorrow. Any questions up there? Yeah?
So what's the difference between what's going on in the green and the most prefrontal area?
Well, in the frontal, right. Well, in the frontal area, you're probably taking all this information in. That seems to be the first place that the information from the parietal association cortices goes. So now you're sorting it out. You're sort of parsing the bits. And then from parsing the bits, you make a plan, kind of a pre-plan, and that goes to the premotor areas. And that sets up, that's the green. And then the premotor areas, that sets up a pattern then that then you can, in short order, activate those upper motor neurons to activate the lower motor neurons. Okay. It gets worse than that because there's a large number of the premotor areas will have descending pathways as kind of upper motor neurons, the synapse into lower motor neurons. But let's not open that can of worms. Let's keep these guys separate. Anything else? Okay, with that, I'll see you again tomorrow.